I'm Attila Haidu. I'm the CEO of Stelvio Therapeutics, and we're developing therapies through epigenetics. What is cancer? Cancer is uncontrolled growth of abnormal cells that don't die. This is what breast cancer looks like, and this is what brain cancer looks like. These are terrible diseases that impact all of our lives. So we started off by asking, if cancer is a developmental disease, can we reverse the process back into normal cells that can die of old age? Cancer stem cells divide symmetrically, which is basically a mother cell creating identical copies of itself. On the other hand, normal stem cells divide asymmetrically, which serves two purposes simultaneously. One is self-renewal, and two is producing generations of differentiated cells that can die. We tackled this problem using epigenetics because epigenetics controls gene expression. There's only two kinds of chromatin. There's open and there's closed. With open chromatin, the DNA is easy to read and the gene is expressed. With closed chromatin, the DNA is hard to read and the gene is not expressed. So we're labeling the histone methyl groups with antibodies, as shown in the orange shape in this diagram. We then invented a novel platform screening technology called microscopic imaging of the epigenetic landscape, or MIEL for short. This is the nucleus of a cell. We've labeled the histone methyl groups with antibodies. Green is closed chromatin, red is open chromatin, and the blue is DNA. So we're segmenting the nucleus and extracting the features such as texture, bumpiness, and gradients. There are over 500 features in all, which gives us a pattern. We then use machine learning to recognize the patterns and assign every single cell a unique epigenetic signature, which is akin to a barcode. Now, the barcodes for a cancer stem cell are completely different than the barcodes for a normal cell. This is the core of our technology, it's cell identity. We can visually see the difference between a cancer cell and a normal cell. Next, you will see a new paradigm in drug screening with live imaging. Watch the cell in the center circle, and you'll see it dividing. This is important for two reasons. One, we can see asymmetric divisions, and two, we can track changes in the epigenetic landscape. Our approach is amenable to high-throughput screening of small molecule libraries. It's universal. It's applicable to many tumor types and other developmental diseases. It's 1,000 times more cost-effective than conventional sequencing. High speed. We can screen 1 million compounds in less than 1 year. But most importantly is the robustness of the epigenetic signatures, which can identify the hits, which is not feasible with other approaches. This is a distance map which takes the machine learning data from 500 dimensions and crunches it into 2 dimensions because that's all we need. So cancer stem cells map close, and normal differentiated cells map far, and here we can see which small molecules induce the reversion of glioblastoma cancer stem cells back to normal cells, as shown in the red and green dots, which map close to the positive controls of serum and BMP4. This is a distance map based on our gene expression data, which confirms our platform. We can see our top hits, in particular topicide, which induce the reversion of glioblastoma cancer stem cells back to normal. We're very excited that Johnson & Johnson have invited us to join JLabs in San Diego starting September 26th. They're also open to using our platform technology to screen their compound library of 1 million compounds. We're also engaged in discussion with Sanofi to identify novel targets and therapeutics that could be used as treatments for neurodegenerative diseases such as Huntington's. So these conversations have opened up using our approach in other developmental diseases such as Alzheimer's. I've successfully executed our business model in my past many times, which involves partnerships with big pharma. We will use our technology to screen their libraries and come up with hits. We will then take these hits and optimize them through medchem, through structure-activity relationship work, and develop an understanding of the mechanism of action. We will then deliver these lead candidates to our pharma partners who will take them forward through clinical trials. In exchange, we'll receive upfront milestone and royalty payments. I'm Attila Heidi. I'm the CEO of Stelvio Therapeutics. I have over 20 years of experience in big pharma in all areas of the business, and I co-founded Stelvio Therapeutics with Alexei Tursky, who's our CSO. He's an associate professor of neuroscience at Sanford Burnham Medical Discovery Institute and an expert in epigenetics. We have a world-class team of advisors with David Andrews, an expert in machine learning, and David Spanner, who's an expert in the tumor microenvironment models. So why are we doing this? We're doing this to deliver medicines of value to the patients that need them. In order to do that, we're raising $4.1 million to operationalize our drug discovery and development efforts. Please join us after the presentations. We'd love to talk to you. Thank you for your attention.